Right. Hello everyone, and welcome to our webinar called Holotomography meets Microfluidics. So my name is Bruno Combet. I am the sales director of Tomocube, and I have the honor to moderate this webinar, in which we will show you the work done by Lucile Mercier at the Gustave Roussy Institute near Paris. And it's a joint webinar between Tomocube and Okomera. And before Lucile starts to discuss her work, let me just show you a couple of slides to explain what holotomography is and what kind of image we can generate with it, because it's a very innovating technology. And it's all about refractive index, actually. So we are making images by measuring the refractive indexes in 3D for the whole sample. And the way we do it, actually, this is the simple law that relates the refractive index and the angle of the light deviation. Refractive index gives you an indication of the density of the material. In the cell, each individual component has a particular refractive index. The nucleus has a refractive index. The nuclear membrane has a refractive index. Nucleoli, mitochondria, all this individual component has a specific content of lipids, proteins, sugar, whatever, that makes a local density. And we can measure this local density with a super high precision of measurement and super high precision of localization. We go down to 160, 180 nanometers lateral resolution. And the way we do it, we use structure light, structure illumination. We create black and white pattern that we project, and then this deformed image is going to the camera. We do this projection with multiple angles, multiple patterns, and at the end we can kind of deconvolve, do a deconvolution, and find the accurate value of refractive index of any position of the cell in 3D. And here it's a nice example of a typical image we can generate, where we can see mitochondria, we can see lipid droplets, nucleus, nucleoli, and all this information. This is typical image we generate. This image has been taken by Lucile. It's a tumoroid, and you can see it's an optical section, because we do 3D, we do optical sectioning, same idea as with a confocal, and we do optical sectioning, and here you can see clearly, you can count the cells, you can see the nucleus, you can see even the nucleoli, you can see mitochondria, you can see a lot of details without any label. So I should have said this is label-free. Just take your sample, put it into our machine, image, and you get such a nice image that you can use and and you can analyze. This is what we are going to show you. This is a picture of the machine that has been used. It's a Tomocube Holotomography HT X1 series, together with the Okomera chip that will be described by Lucile, where you can do, you see, all these little wells where you can combine drugs for a tumoroid. And this system is fully compatible with off-the-shelf plate and plate holders. All right, so I think now the stage is for Lucile. I stop sharing, and Lucile, you can share your.
Thank you, Bruno.
Before you said, I should have said, you have the chat, the question and answer chat session, where feel free to ask your questions, and we'll be answering the question right after the session.
Okay. So thank you, Bruno and Jory, for inviting me to this webinar. It's a real pleasure. So I'm going to present you today very briefly the technology developed by Okomera and the results that I was able to obtain using this technology during my PhD. So first of all, just for a bit of context, in terms of statistics about cancer, nowadays it's 1 in 3 person who's diagnosed with cancer, and more than 200 drugs, cancer drugs available on the market. However, more than 50% of the cancer patients do not respond or stop responding to standard treatments. It is also important to know that the cancer disease is not just an accumulation of malignant cells. Now, it has been well described in the literature. It is a complex and dynamic ecosystem where the tumor cells actually communicate with their microenvironment, influencing immune escape, drug resistance, heterogeneity, migration. And capturing this dialogue is essential for predicting treatment outcomes. So this is why functional precision medicine aims to directly test drug efficacy in ex vivo condition using cells freshly obtained from patient biopsies. For this approach, to be clinically meaningful, the experimental model has to meet several key requirements. It must be compatible with very limited amount of biological material that is available after a biopsy or surgical resection, for instance. Which makes here traditional culture systems not compatible with such low input, as it often requires bigger sample or sample expansion phase. Secondly, the readouts should be usable within a clinically relevant timeframe, so it can really be involved in the therapeutic decision making. And finally, it has to preserve the tumor heterogeneity and complexity. This means not only maintaining the epithelial tumor compartment, but also keeping some of the stromal populations that critically influence the treatment response. So here, the solution developed by Okomera address this problematic. So as you can see here, we have a representation of our microfluidic chips. So during my PhD, I have developed a workflow that starts from the surgery on day 0, where the surgeon gives the tumor piece to the pathologist that sample a piece of the tumor and give it to me to the lab where I process it. So I start from this little piece of tumor that I dissociated using mechanical and enzymatic dissociation to obtain a single cell suspension. This single cell suspension will remove all the immune fraction and keep only the stromal and the cancer fraction, the tumoral cells and the stromal cells. So this heterogeneous cell suspension will be encapsulated within droplets using our technology. And the droplets will be injected into our chip using our machine. The cells will then sedimentate into the bottom of the droplets, and we put the microfluidic chip in the incubator. And after 48 hours, the microtumors are formed. Once those microtumors are formed, we are going to produce and inject a library of drugs. So here, as you can see, each droplet contains a single condition that is associated with a fluorescent barcode that enable us to recognize the different condition injected. And so here, the secondary droplet will get trapped in the secondary trap, and the two droplets will be merged together to really inject the treatment on the tumoroid. To follow the effect of the treatment, we use microscopy readout associated with also fluorescent microscopy to follow the evolution of the fluorescent marker, for example, for death marker, for instance. And finally, we have developed our own analysis software based on AI algorithm, that enables the analysis of all the single tumoroids present in the chips. So using our platform, we are able to generate large amounts of uniform microtumors from patient biopsies, ensuring a strong reproducibility across independent trends and between the different samples. As you can see here, on these 8 different patient samples, we're able to have similar diameter of the different tumoroids and hundreds of replicates of tumoroids. Such scalability is really essential for functional precision medicine that enables parallel drug testing on standardized microtumor units and producing results that are both robust and clinically actionable. So by using our drug library, I was able to test multiple therapies on microtumor. Here, for this case, on one patient, I directly compare 3 drugs and their efficacy to pinpoint really the most promising treatment for that individual. So here I have tested an active metabolite of cyclophosphamide, 5-FU, carboplatin. And in this case, for example, we can see that the carboplatin is the most efficient drug among these 3. And the second approach is to test a single drug across a cohort of 10 patients. And here, in this case, we found differential sensitivities, as you can see, highlighting here the interpatient variability and informing which subgroups can actually benefit the most from that specific therapy. So the platform, the Okomera platform being fully compatible with immunofluorescent assays, I was able to characterize the tumoroids directly on chip, allowing to highlight the cellular heterogeneity and structural organization of the microtumor. As you can see here with these 2 markers, the cytokeratin 8 and 5, we are able to detect the different subpopulation of cancer cells. And here, using the actin, the filamentous actin marker, we are able to really assess the organization of the tumoroid. So while this approach is quite rich in phenotypic information, it remains destructive assays that prevent the follow-up analysis on the same tumoroid over time. Oops, sorry. So as I mentioned, these organoids generated are complex structure composed of stromal and cancer cells so with multiple cell types. But with the current endpoint assays of immunofluorescence, for instance, we can only capture static snapshots. So we cannot continuously quantify the stromal versus the tumoral zone or follow how this region reorganize during the treatment. So tracking the spatial and temporal dynamics remains really key challenge that limits our ability to fully interpret the therapy response. So this is where Tomocube technology bring a real added value to the Okomera platform. So thanks to the broad sample compatibility of holotomography, we were able to acquire images of our chips within this system. So that enables us to acquire multiple chips on the same run. And the field of view size was large enough to capture the biopsy-derived organoids, which can reach up to a diameter up to 150 micrometers without any stitching. So in one shot, we have an entire tumoroids acquired. So as you can see here on the left, the holotomography shows us really rich morphological information. As you can see, we can detect the single cell that are composing the microstructure. We can see the nucleus, the nucleoli also, and the granules, for example, you can see here and here. So this is only based on the refractive index difference. And here we can also track it over time without any damage, without any phototoxicity. So here we expected that we might be able to find a novel phenotypic marker to differentiate this heterogeneous microstructure, and not in the endpoint, but in early stage of the drug treatment, and to follow it up during the whole assay. So the first challenge was to find a feature that differs from the tumoral region to the stromal region of our tumoroids. Because in fact, based on the immunofluorescent staining, we were able to identify 2 regions on the tumoroids. The tumor region that is characterized by the expression of cytokeratin 8 and cytokeratin 5, as you can see here, and the stromal region that is negative for the expression of those two cytokeratin, as you can see right here. And when combining these immunofluorescent images with the holotomography images, we found the presence of small granules, highly refractive, especially in the stromal region. We were able to quantify them thanks to the TomoAnalysis pipeline that Joey will describe it to you just after. And the small vesicles can very likely reflect the secretive activity of the fibroblasts found in the stromal region. And we can see here that by quantifying it using the TomoAnalysis pipeline, we really have these significant results of high granularity in the stromal region compared to the tumor region. So we use this metric, the granularity, to split all the tumoroids in one chip into two subgroups. One subgroup with a low global granularity. So basically tumoroids that contain smaller stromal regions, and a second group with high granularity that contains bigger stromal regions. And what we were able to see is that the range of delta volume between before the treatment and after the treatment, so the delta volume range was much smaller in the high granularity subgroup, meaning that the tumoroids with higher contents, higher stromal contents, might be less sensitive to the carboplatin. And this is what we can see on our different patients, that in fact, in some cases, we are not able to reach the 100% maximum cytotoxic efficacy. And this could be very, very likely either resistant subpopulation remaining or also eventually residual fibrosis remaining after the treatment. And so this is why this data really reinforce the idea that stromal architecture is a key determinant of the therapeutic outcome and must be taken into account in ex vivo drug testing. So now I'm going to let Joey explain you all the background of these results with all the analysis pipeline.
Hello, this is second presenter, Joey Lee. So I'm going to introduce about how we created the insight from the holotomography images using the TomoAnalysis, which is our analysis software from the Tomocube. So let's start with the motivation we got. So during the time lapse, as you can see in the Lucile's presentation, during the treatment, the organoid region gets separated into two parts, and surprisingly, the two part has like the distinct phenotypic difference using the granule density. So after the treatment, we stained with the immunofluorescence using the tumor markers, and those regions with the high granularity was negative to the tumor markers, and those regions with low granularity was tumor marker positive. So based on this observation, the difference in granularity were observed between the positive and negative regions within the patient-derived organoids. So we raised two questions. Is this granularity can be an indicator of the stromal contents? And also, if so, can this granularity predict the sensitivity to the drug? So let's go for the details for this first question. So I said granularity, but how can we calculate the granularity? Normally, we define the granularity as the density of granule-like structures in defined region, for example, within a cell or organoid. So basically, we take the number of granule or volume of granule and divided it into number of cell or volume of organoid. So in this case, what do we need to calculate the granularity? So the volume of organoid, which will require the segmentation of the organoid or each region for tumor or stroma, and the counting for the cells, And definitely we will need the segmentation for the granule to count or calculate the volume. So for the analysis schematics, we take the holotomography and put the images within the TomoAnalysis with our custom analysis pipeline. And then it will process the images with our defined algorithm, and it will create the segmentation and the calculation result as well. So as a result, we will be able to create the segmentation result and the features that we will need to calculate the granularity. So first, for the organoid and region segmentation, so I used the pre-designed pipeline within the TomoAnalysis for organoid segmentation and the pipeline for the fluorescent object segmentation. So the features in the holotomography is taken to segment the whole organoid, and the fluorescent image, especially for the tumor markers, are utilized to segment the tumor positive region. And then I just subtracted the tumor region from the organoid to take the negative region, which means the stroma region. And then also using the fluorescent segmentation pipeline in the TomoAnalysis, I just put the DAPI staining as input, and I was able to create the counting for the cells as well. And for the granules, so here actually what we call as granule in the organoid was like sharing the similar morphologies and characteristics with the lipid drumlet in the common adherent cells. So I was able to utilize the lipid drumlet segmentation pipeline in the TomoAnalysis to segment the granules. So in summary, I was able to calculate the region volume for stroma and tumor, and count volume and dry mass were measured for the granules as well, and counting for the cell. So by making some combination of these features, I was able to calculate the granularity and compare the tumor and stroma region. And the result was, in all combination, the stroma region exhibited the higher granularity compared to the tumor. So in summary, we got the first answer. So is the granularity can serve as an indicator of stroma contents? So the answer was yes. So if so, can this granularity can really predict the sensitivity to the drug? So to answer this question, we measured the granularity in the real-time data as well. So actually in this case, as you know, it is impossible to apply the immunofluorescence in the live organoid, even though there is some exceptions. So basically what I did here is segment the granules and segment the organoid with only the holotomography, because we don't have the fluorescence here. So the scheme was just the same. We take the holotomography and put in the TomoAnalysis with our custom analysis pipeline, and we got the granule segmentation. And here for the organoid segmentation, I was also utilized the microsem as well. So as a result, we were able to divide the whole organoid into two groups. One group has high granularity and another group has lower granularity. So based on the assumption that tumor has lower granularity, the organoid within the high granularity group has the lower tumor fraction within there, and the organoid in the low granularity has higher tumor fraction. So as we all know, normally the anti-cancer drugs primarily target the tumor cells, not the stroma cells. So higher tumor contents normally leads to the stronger drug response. So as a result, in the high granularity group, even though we treated the carboplatin, the anti-cancer drug, with the most strong dose, the volume is not changed from the before we treat the drug and after we treat the drug. But in comparison, in the low granularity group, even though we treated the carboplatin in the middle concentration, they reacted a lot compared to the high granularity. So cells were dying and the volume decreased dramatically. So as a conclusion, we answered the two questions. Is the granularity can serve as an indicator of stromal contents? Yes. And can this granularity predict the sensitivity to the drug? Also, yes. So we found that the granularity is a key determinant of the drug response within the heterogeneous patient-derived organoid. So before we end, I would like to emphasize some benefits we got from this workflow. So the first one is the full customizability for diverse analysis target, like this kind of like special granules within organoid. So basically, in TomoAnalysis, we got 3 pre-designed pipeline, which we utilized in this case: the organoid segmentation and the fluorescence segmentation and the lipid droplet. So as a result, we were able to segment the organoid and the tumor region and the granules. And on top of that, the organoid and granules came from the holotomography, which means we didn't apply any staining on here. And also, the holotomography and fluorescence can be acquired at the same time. So basically, we can take the phenotypic features only with the holotomography, and additionally, we can also utilize the information from the fluorescence as well. So in summary, TomoAnalysis turns your images into data and intuition into reproducible analysis. What you can see is what you can believe, and if you can measure, you can prove. So this is the end of our webinar. Thank you for your attention, and let's move on to the Q&A session.
Thank you very much, Joyce. So we have a good number of questions already asked in the chat box. I had also some questions sent via a private channel. So we can start with an interesting question. I think that's for you, Lucile. Question from Andrea asking, how can we make sure that only one droplet with a drug is fused with a droplet that contains the tumoroid? Can you answer this?
Yeah, sure. So actually, the design of our chip enables effectively the trap of only one droplet, either primary or secondary. If the droplet has the right size, only a single droplet will be trapped in our chip. So it's really the specific of our design, and that enables really the control of the condition that we have in our droplets.
Good, that's clear. I had someone asking me, if we buy the machine, do we need assistance for the image analysis, and can you help us? So Joey, can you comment on this?
Yes. So we provide 2 kinds of license for TomoAnalysis, and we also have the subscription license as well. In the subscription, we also do have this technical support, those who purchase the subscription models. So you will be able to get the technical support from the Tomocube as well.
Good. And while you're here, there is a very technical question. How long does it take to perform the analysis of 10,000 spheroids with Tomocube? And how can metabolically active cells be distinguished from non-active cells? That's Yasin Bunhab that is asking this. Can you answer this? At least a rough idea of the time-consuming analysis. And not sure I understand really metabolically against non-active cells, but please, Joy, explain us.
So actually, in the analysis part, it depends on what kind of organelles or features are you interested in. And also, we also have the acquisition as well. So for the acquisition, I think you can take 2 minutes for like 1 minute for each spheroid. So you can get some rough calculation for the acquisition. And also, I think for the analysis, you can take 2 or 3 minutes for a single FOV. So based on just simple calculation, I think you can get some idea for the rough calculation for acquisition and the analysis as well.
Okay, so we try to give quicker answer because now questions are coming. We're
Yeah.
snowed under. How do you handle the fluidics in the machine? We have incubation chamber with little holes, and then we can pass the tubes through the holes in the chamber, and we can handle this microfluidics with peristatic pump or whatever, or the Okomera system. This is fully compatible. And why the refractive index is always between 1.3, 1.4. It's not always the case. Usually it's between 1.33, which is close to the buffer or the let's say very aqueous things, up to it can go up to 1.45. Our precision is 4 digits down to the after the comma. So we have like 9,999 level as a dynamic, let's say like this. That's a super high precision. And again, remember the localization, the lateral precision is around 160 nanometers. We ask also what kind of organoids are you growing? So we have publications with a lot of different type of organoids that you can grow. Lucile, can you just list the different types you've done?
In terms of medium or type of organoid?
No, of organs.
Actually, we have several. We have colorectal, we have pancreatic cancer. So in my case, it was breast cancer, different cell lines, different PDX as well. So we have tested already different type of organs yeah.
And have you tried non-human cells?
Non-human cells, yeah, we
Yeah.
have tested already some cell lines, murine cell lines, actually.
Okay.
Mm.
Okay. And the medium, what kind of medium you're using?
So this is really
Like
dependent on the type of organoids you're using. We are trying to reference to the guidelines, the recommended guidelines. So yeah, it's really depending on the model you're using.
So interesting question. Do you have any artifacts that occur towards the deeper end of the capture of the tumors? The answer is yes. The further you go from the objective, of course, the more, let's say, chromatic and spherical aberration you get. So yeah, that could be some artifacts. I can phrase it like this: this is not artifact-free technology, but our algorithm is improving and improving again and again, and so we make sure that we have less artifacts that we can. And how do you validate that the model resembles patient behavior close enough? So I don't know if Lucille, you can comment on this or
So
any idea.
this
That's a
is
very interesting question. Thank you,
yeah. So
Yannick.
it's preliminary results so far, but validation are ongoing, especially, for example, through sequencing. We are also capable of collecting the organoids after culture, to collect it and, for example, to extract DNA or RNA from the organoids. So this is also compatible with the technology, and also through immunofluorescent stainings, for instance. So there are different application that we can do as endpoints.
Okay. So there is a question from Amir that's easy to answer. How good is, when you do stitching, how good is the refractive index measurement in the overlap region? Actually, we don't have to overlap, and the stitching is absolutely perfect. And there is a good reason for that, is that we measure numbers. We don't have this illumination shadow that you have when you stitch with regular conventional microscopy. we just stitch numbers, and the refractive index, either the sample is on the left or on the right of the field of view, is absolutely the same to the super high precision. So the stitching is 100% perfect. You can't tell how many tiles you've stitched after the stitching. How does a media with a very refractive index affect the tomogram quality? Yeah, I mean, that's also another good question. It does affect, of course, if you have an enormous mismatch, like if you use a super refractive oil, these kind of things. So yeah, I mean, these are some, let's say, pitfalls or, I mean, drawbacks, let's say, of the technology. But most of the time, let's say 99.9%, all culture medium, even for IVF, the ovoid or this kind of things is fully compatible with our technology.
And if I can complete your
Yeah,
response also,
sure.
Bruno, here in the context of the microfluidic chips, we are in a context of emulsion. So the droplet is aqueous, so it's the culture medium that contains the cells, and surrounding that it's the oil. And in this emulsion context, we didn't had encounter any issue concerning the refractive index on our acquisition. So far, so good.
So far, so good. So because we do have so many questions here, we can't answer. I mean, we'll take few more, but here you can see contact email addresses. So feel free to email me if you have questions that I haven't answered, or Sumin or Okomera people if you think it's more like for Okomera, or both if you want. Yannick is asking, is the droplet location random? Lucile, can you comment on this?
Yes, it is. It is random.
So it is random. So basically, you're using the color to see.
Yes,
Okay.
exactly. We determine we have a specific barcode. Each droplet is associated to a specific dye, fluorescent dye. And thanks to the fluorescent imaging, we acquire the whole barcode and we are able to really recognize what condition is in which droplets.
Okay. Ron Zhao has asked a very interesting question. Is it possible to change objective to get higher magnification? Actually, Ron, you don't need higher magnification because we already have such a good resolution. The way we get such good resolution is because we are making synthetic numerical aperture. We're using angles for the light, so we force the objective lens to to increase the angle of collection of the light, and therefore we generate higher numerical aperture. And the resolution actually is so good that you don't need to go like 100x magnification. You're not going to increase the resolution. The pixel size is around 120 nanometers, and that's already a super good pixel size that you don't need higher magnification. So no, we don't give you the chance to change objective because you don't have to. You don't need it. So what do we have? Joey, that's for you. Is the calculation based on 3D reconstruction or on 2D slices? How to avoid overcalculated between slices?
So yeah, the
Yeah.
calculation is based on the 3D in this case. So actually, we can decide whether we go for 3D or 2D based on which information we need for the analysis. So some cases, like counting the cell, doesn't have to be necessarily in 3D. You can also get enough numbers in the 2D as well. So
Okay.
yeah.
Yeah, go ahead.
No, yeah, that's it.
You're done. Okay, so Alberto is asking, I saw that one of the images had the granule labeled unknown granules. So that's a good question because sometimes you don't know what you're seeing. You see everything, but could be lysosomes, as you say, could be reticulum, something. So to discriminate between the structures, you need to use the specific dye with fluorescence, like mitotracker, lysotracker, or anti-lipid to check whether it's a lipid droplet. With experience, we know when we see the refractive index value, we see the size, we see the localization in the cell and so on, we can guess. Lipid droplets, there is no ambiguity. We know where they are. We know how they look like. Sometimes, vesicles, Lysosomes, it's difficult because they may change also their structure depending on the cell condition and so on. So yeah, to answer the question, you need a specific probe, and then you look at the image in fluorescence, and then you look below the image without the fluorescence, say, oh yeah, that's what I'm looking for. So that's the way we can answer the question: is it lysosome or another vesicle or something else? So, Lucile, one of the last questions for you is regarding the Okomera chip. Is this secure, suited for applications that need containment, like infectious material? Or in other terms, can you use the Okomera chip with infected cells?
Yeah, so actually, we have already performed some and published some results about the CRISPR-Cas9 applications and lentivirus. So typically, this is an example. So we can use lentiviruses within our chips, for example, encapsulated in the droplets. And so I invite you to look at in our website the different posters that we have with the different results that we shared already in conferences.
Super, thank you, Lucille. Lisa is saying first, thank you for the amazing presentation and research. Thank you, Lisa. When will Tomocube address the clinical for embryos or ovocytes? We're doing it. We're on it. We're on it. We have a very strong collaboration with IVF clinics all over the world, and we are doing a lot of advanced research on embryos and ovocytes for assessment and so on, because holotomography is absolutely the perfect tool to really see everything that can give you information on the size of the embryo, how many cells, and so on, and ovocytes also to measure to see the state of the nucleus, the zone pellucid, and all this. So we're on it. We're on it. So it'll come very soon. Yeah, I think there must be some unanswered question, but okay, a last one for Joey. For holotomography, how to create the pipeline to distinguish the unlabeled cell which contains more than one cell type if the refractive index of the cell overlaps.
So based on the features, for example, after the pipeline, we got the segmentation and the measurement for the phenotypic features for each cell lines. So using the difference within the phenotypic features for single cell line and another cell line, we will be able to distinguish those two cell lines.
Okay. So I would love to thank you very, very much for your presence here, for your time you spent. This webinar has been recorded, so you'll have access on both websites to the recordings. If you have any additional questions, just contact us. I gave you the email addresses, but we're available on all social media and so on, so easy to find. Thank you again, and I wish you a good rest of the day or the night or the morning, wherever you are. And thank you very much. Goodbye.
Thank you. Bye-bye.
Thank you.
Bye.